First, are there any questions or concerns? You'll see we're a little behind. And I see that it's got the chapter 12 information due. I'm going to extend it out. Let me think about how far to extend it out, but realize that we're just starting chapter 7. It's going to start going a lot faster. We're going to start going through chapters faster. However, I was just looking, bear with me. It says that chapter 12 is due on the 27th. We won't have it done by the 27th. So I'm going to extend it out at least another week. Any questions? So we're starting chapter 4 today. I'm sorry, chapter 7 tonight. Have you talked about bone in lab yet? You have?
Yeah, we have.
Yes, yes.
Okay, so some of it is a review for you, which is not bad. So the skeletal system consists of bones, cartilage, and ligaments tightly joined together. Cartilage is the forerunner or the precursor of most bones in the embryo skeleton, and it's involved in bone development in childhood. It also covers the joint surfaces in the mature skeletons. Ligaments hold bones to bones together at joints. Tendons attach muscles to bones. Of the skeletal system, bone, cartilage, and ligaments are the 3 different types of structures that we have in the skeletal system. Tendons are not included because they are muscles to bones. Now, this is kind of a trick question because we haven't answered it yet, but which of the following tissues could be found on or within a bone, such as a humerus?
Want to be all of them?
All of them.
How did you know that?
Because of
I
lab.
just started reading chapter 7.
You're right. You're right.
Well, it says tissues, and all of them are tissues, so it was a good guess.
Okay. Well, all right. Yes, you're right. Okay. The skeleton it's a composition of multiple tissues, which include nervous tissue, blood, adipose tissue, which is yellow bone marrow, dense regular connective tissue, and dense irregular connective tissue. And so forth. And it also has reticular connective tissue, which is, if you remember, that's one of the lymphoid organs. Talk about why you find it in bone here in a moment. Also, bones continually remodel themselves and interact physiologically with all the other organ systems. We often think of bones as being stagnant, but they're anything but. They're constantly being broken down and remodeled. Therefore, we can think of each individual bone as being an organ, because each bone is composed of multiple tissues that are constantly in a state of dynamic flux. Some different functions of the skeleton is that the skeleton is there to support the body, teeth, viscera, or organs. It supports soft tissue and provides attachment points for tendons of skeletal muscle. It's involved in protecting and enclosing internal organs. It's involved in movement using the bones as levers. Muscles allow for movement and breathing and so forth. Also, bones are involved in balancing electrolytes through their storage and a release of calcium and phosphate minerals. It also functions in the acid-base balance through buffering the blood against excessive, remember the acid-base meaning pH. So against excessive pH changes by absorbing and releasing alkaline phosphates and carbonates that can work as weak acids and weak bases. Remember how a buffer is a combination of a weak acid and a weak base? So through the release of calcium and carbonate salts, calcium salts, we can allow for the minerals in skeleton to work as weak acids and bases. Also, the skeleton is involved in blood formation because the red bone marrow is where the blood and immune cells are produced. So a lot of the red bone marrow is composed, or the majority of it is composed of hematopoietic tissue, which is a tissue where new blood cells are formed. Some vocab terminology or some words to remember, some base words, root words to remember. Hema means blood. Hematopoietic or autopoietic. Let's leave it this way. Let's say poetic. Means a progenitor cell. Something that's going to become something else. It's actively dividing and it's going to become something else. Progenitor or precursor. Precursor. So the red bone marrow contains hematopoietic tissue, which is where new blood cells are formed. I have 3 different types of formed cells from hematopoietic tissue. We have platelets, red blood cells, and white blood cells. Platelets are involved in blood clotting. Red blood cells, remember this is oxygen and carbon dioxide transport. White blood cells are the immune system. Which of the following functions of the skeletal system would be considered a metabolic function?
D, movement.
You're close. Metabolic,
Storage.
it's actually mineral storage. Because if you think about it, how all the reactions are going to require different minerals. So we have four basic types of bones. We have long bones, we have short bones, flat bones, and irregular bones. An example of a long bone would be your humerus. Short bones are your carpals and your tarsals. Flat bones, like the bones of the cranium or the sternum. Irregular bones are ones that can't be grouped into any of the other three groups. So they're like the vertebrae. Or another one might be the coxa, the os coxa of the pelvis. All bones have an outer layer of periosteum. Osteum always means bone. Peri always means around or on the periphery, surrounding periphery. So periosteum is the layer, is a tissue that's surrounding the periphery of the bone. Endosteum, endo meaning the internal surface, osteum, so this is the inner layer that covers the internal bone. Red bone marrow is found in the internal surfaces of the bone, in the internal cavities. So all bone, not just flat bone, but all bone contains an outer surface of compact bone and then an inner spongy bone or cancellous bone. Looking at the basic anatomy of long bones, compact cortical bone outlines the whole entire surface. This compact bone encloses the marrow or the medullary cavity that contains the bone marrow. Marrow mean because we call it the marrow cavity because it contains the yellow or the red bone marrow. We can also, it's also referred to as the medullary cavity. Medullary meaning internal area, internal space, internal or inner or internal. So this is the area inside the bone. We're going to see this again. It actually, medulla means inner or internal. So medullary cavity is the internal cavity. Spongy or cancellous bone is in the central space internally and lines the medullary cavity. So it's found at the ends of the bone and it lines the inner cavity, the medullary cavity. In additionally, the ends of the bones are called the epiphyses. Epiphyses. The diaphysis is the shaft or the middle part between the epiphyses. So epiphyses are the distal and proximal ends of the long bone. The diaphysis is the inner shaft area. The metaphysis is where the epiphyses and the diaphysis meet up. Lots of physes words here. So epiphysis, metaphysis, diaphysis. The epiphyseal growth plate is where hyaline cartilage in the metaphysis allows for growth of the long bones. So we're going to find this in the metaphysis area where the epiphyses meet up with the diaphyses. Remember that it's hyaline cartilage. Then you have different types of cartilage, the fibrocartilage, the elastic cartilage. This is a hyaline cartilage. Articular cartilage is the layer of hyaline cartilage covering the regions of the epiphyses where the bone articulates with other bones. So where in the long bones is hyaline cartilage found?
Isn't it on the ends?
It's on the ends. Where else is it?
In the epiphysis,
Yep.
in aging children.
Yep, in the epiphyseal plate. Once the cartilage in the epiphyseal plate is depleted, the bones will stop growing. And then an epiphyseal line will form in that area. So the medullary cavity is the space within the diaphysis that in adults or reasonably adult children contains yellow bone marrow. The yellow bone marrow is a fatty tissue that acts as a reserve fuel supply. The endosteum is a layer, remember endo meaning internal, osteum meaning bone. So this is a layer of reticular connective tissue that lines the inner marrow cavity. And why is it reticular tissue? Well, if this is the marrow part of the bone, which is involved in lymphoid and lymphatic, or the lymphatic system. It's a good idea, or it's a good connection to say that we're going to have reticular connective tissue lining the internal part of the bone where the red bone marrow is, that's involved in the lymphatic system. The endosteum also has cells that are going to dissolve the osseous tissue and others that are going to build it or deposit it. We also have nutrient foramina, these tiny holes where blood vessels are going to enter the bone. Foramina means holes. So this is where blood vessels and nerves are going to enter the bone. Periosteum is the sheath of tough fibrous collagenous layer and an inner osteogenic layer of bone forming cells. So this is tough, tough, dense, irregular connective tissue with an inner layer of osteogenic cells. Genic so osteo means bone, osteo means bone. Genic means is a precursor for cells that can be formed, or it's a word for it, osteogenic, creating. So osteogenic is something that's going to create bone. Some of the collagen fibers of the periosteum are continuous with tendons. Remember, tendons are attach muscles to bone. Some of the collagen fibers of the periosteum are perforating fibers called Sharpie's fibers that are going to penetrate the bony matrix. Any questions so far? So general features of flat bones, they have that sandwich-like construction. There's two layers of compact cortical bone enclosing a middle layer of spongy or cancellous bone. Both outer surfaces are covered with periosteum. The inner spongy layer is in flat bones is called diploie. It helps absorb shock, and the marrow spaces are lined with endosteum. Bones are made mostly of osseous tissue. The connective tissue in the matrix, which is called osteoid, is hardened via mineralization by depositing calcium phosphate and other minerals onto that connective tissue. So osseous tissue is a connective tissue that contains an extracellular matrix surrounding widely separated cells that are living in those lacunae. Sorry, we haven't talked about that unless you've talked about it in a lab. widely separated cells. The matrix components are 15% water, 30% collagen, and 55% crystallized inorganic mineral salts. The crystallized calcium phosphate salt that's part of this inorganic mineral salts is hydroxyapatite. Calcification is the process by which osteoblasts initiate the deposition of mineral salts in a framework of collagen fibers. Leo, you can't be right there. Nobody. So adding some more vocabulary. Any time that you see the word blast on the end of a cell, that means a building cell or a secreting cell, building or secreting cell. Anytime that you see the word classed on the end of a cell, on the end of a word, that is a cell that is breaking down type of, a cell that's involved in breaking down. Or destruct, let's say, cell that breaks down things. Stuff, breaks down stuff. Anytime you see the word site on the end of a word, that is a cell that is essentially in a state of just maintenance, keeping things together by maintaining. So a cell involved in maintenance. So an osteoblast is a cell that's involved in building and secreting bony matrix. An osteoclast is a cell that breaks down bone. An osteocyte is a maintaining bone cell. The principal component of bone that contributes to its hardness is what?
Is it A?
Say it again.
Is it A?
It is, it's A, hydroxyapatite. So bone is a connective tissue that consists of cells, fibers, and ground substance. The four principal types of bone cells are osteogenic cells. Remember, being precursor cells, genic meaning they're going to become bone cells. Osteoblasts, osteocytes, and osteoclasts. Osteogenic cells are continually multiplying stem cells that arise from embryonic mesenchyme. Mesenchyme is the embryonic, undifferentiated connective tissue. embryonic, undifferentiated. So it's not, it's connective tissue that hasn't specialized to become a particular connective tissue yet. Undifferentiated or unspecialized. Connective tissue. So these are stem cells that are rising from embryonic mesenchyme that are going to produce other bone cells. They're present in the endosteum and in the inner layer of the periosteum. Osteoblasts are bone-forming cells that undergo osteogenesis. So genesis, remember, if we talked about the word genesis? Genesis means, if you think it's probably the easiest way to remember Genesis is if you're familiar with the Bible. What's the first chapter of the Bible?
Genesis.
Genesis,
What does Genesis mean?
the beginning.
What
Newly
else can it mean?
Newly forming. Or
Yes,
Earth.
or creating. It's where the
Creating.
creation of the world occurs in the Bible. So Genesis always means the creation or creating things. Creating things. Creating. So osteoblasts are going to undergo osteogenesis, meaning they're producing the organic material of bone and promoting the mineralization of that osteoid organic material. They don't divide, but originate only from osteogenic cells, the stem cells. Bone stress or fractures can stimulate mitosis of osteogenic cells that result in increased amounts of osteoblasts being produced. Osteocytes are former osteoblasts that are trapped in lacunae. Lacunae are cavities. Lacunae are cavities. So osteocytes are former osteoblasts that are trapped in lacunae within the matrix that they deposited. So osteoblasts secrete a bunch of matrix. That's what they do, is they just keep secreting matrix. And then they find themselves trapped in the matrix they've secreted. And at that point, they can't keep secreting more. And so they're trapped in the cavities that are left when they realize they've trap themselves. Let me give you a good analogy of this. You're painting a room, and we'll make it really hard paint. We'll make it red paint, which bright red paint. You're painting a room, you're painting the ceiling, you're painting the floor, painting the walls, and you just keep painting and painting and painting and painting some more. And all of a sudden you realize that you've painted yourself into a corner of that room. And you didn't make sure that you had a way out. And so now you're trapped by all the red paint that you've put down, all the red wet paint that you put down. That's an osteocyte. So an osteoblast keeps secreting matrix until they've trapped themselves in lacunae. And at that point, they become an osteocyte because there's nowhere else for them to keep secreting matrix. So osteoblasts are going to secrete until they're stuck. And then they're going to become osteocytes. If they get unstuck, because bone gets broken down, if we break down the lacunae that they're stuck in, the cavity or the corner of the room, they can start secreting matrix again and become osteoblasts again. That's the best analogy I have for explaining the relationship between those. There are in between, because they're kind of stuck in this little cavity, there are processes by which the osteocytes can contact the processes of other osteocytes outside of their cavities and pass along nutrients and chemical signals and eliminate waste. So they communicate with other osteocytes via these little canals, caniculi meaning little tiny canals. And we'll see those again here in a moment. The major functions of osteocytes is to contribute to the maintenance of bone density and blood concentrations of calcium and phosphate. They are strain sensors that allow for the flow of extracellular fluid in the lacunae and caniculi. They stimulate osteocytes to the strain sensors, meaning increased fluid in the lacunae and the caniculi, will stimulate osteocytes to secrete signals that will regulate more bone remodeling, causing changes in the bone shape and density. So they regulate whether or not bones are broken down or built up. They're maintaining things. They're stuck, but they also are involved in maintaining and regulating whether the surrounding bone is produced, synthesized, or broken down. Any questions? Osteoclasts in comparison, they don't come from the osteogenic cells. So osteoblasts become osteocytes. Osteoblasts come from osteogenic cells. Osteoclasts are actually formed from the fusion of multiple bone marrow stem cells. Remember, those are the hematopoietic cells. So osteoclasts come from bone marrow stem cells or hematopoietic cells. Osteoclasts are bone dissolving cells on the surface that promote osteolysis. Lysis, just like we saw this word in chapter 2. Lysis means to break down. So osteolysis means to break down bone. So osteoclasts promote osteolysis, breaking down the bone. They have different, their structure's a little different. They have a ruffled border. Ruffled border. And the ruffled border is going to increase the efficiency. So they bind to the bone and create this little resorption bay area. Resorption bay area. And the main purpose of that ruffled border is to give more surface area to the cell and allow for more function, more abilities to interact with the bone. So just like microvilli are in the small intestines to increase the surface area, this ruffled border is to increase the surface area. Most osteoclasts reside in resorption bays that they carve into the bone. So this little bay, it's essentially carved it out. Here's the compact bone. Which represents the correct sequence of development of bone cells.
Is it D?
I think you're right. Osteogenic to osteoblast to osteocytes. Which of the following bone cell types is primarily responsible for initiating ossification of the bone?
Is it A? Because they turn into the osteocytes.
It's A. That's right. So blast meaning the building. So it's initiating the building of the bone. Chondro, that's another word. Chondro means cartilage. Chondro means cartilage. So this is chondroblast is a cartilage building cell. So the bony matrix as a whole is made of 1/3 organic matter, which is collagen, protein carbohydrate complexes, which includes aminoglycans, proteoglycans. Remember all this aminoglycan, every time you see the word glycan, you know it's a carbohydrate. Proteoglycan, meaning a protein carbohydrate complex. Glycoprotein, meaning a carbohydrate protein complex. The other 2/3 of bone is inorganic matter. 85% of it is hydroxyapatite. 10% of it is calcium carbonate. And 5% is other ions. The ceramic polymer, so the polymer, the organic matter, and the ceramic, meaning the minerals, combine the optimal properties, resulting in great strength with flexibility. Oh, I have something. 1 second here. I'm lucky I have a supportive roommate who tolerates some of my experiments and doesn't kill me. She's about ready for me to get rid of this. But what this is, and I'm gonna move my computer a little back so that I don't get stuck on my laptop. I took a chicken bone and I soaked it in vinegar for it's actually been in here about 2 months. It doesn't stink because it's a really good preservative. pH is a really good preservative. But I have a chicken bone in here that's been soaking in vinegar for about 2 months. And normally bones are fairly hard, right? Well, this bone's been soaking in vinegar, and if you look at it, I can almost flex it all the way down. I can tell your dog's really interested, Caitlin. He's not sure what to think of this.
I think she heard chicken and she just looked.
But if you look at it, it's really, really, really flexible. The vinegar dissolves out all the inorganic components, leaving only the organic flexible collagen material. So if your bones are struggling with levels of calcium, they become more flexible, but they don't have the same strength and rigidity that they need to have. I haven't done this yet. This will be next time, but you can also cook a chicken bone for a very long period of time at a very high heat. And bear with me, I gotta wipe my hands. Sorry, I had to get that off my hand before I'm playing with my computer again. If you cook a bone for a really long period of time, you can get rid of the organic components. And what you're left with is just the crystallized hydroxyapatite. That will be my next experiment. I'm not going to do that just yet. And so the organic material gives it the flexibility. The inorganic material gives it the strength. So bones that are deficient in calcium are too flexible and rubbery, like you saw with that one that I had. From what I've been told, if you have a long enough bone that you've soaked in vinegar, you could actually tie it into a knot. So bones deficient, this is the basis behind rickets, is where the bones do not have enough calcium in them. Bones that are deficient in protein are excessively brittle. Excessively brittle. What this means is that they tend to break easily. This is the basis of osteogenesis imperfecta, a genetic disorder. So the collagen provides great strength through being able to break bonds that hold bones together under pressure and that can reform afterwards. This is lost in the disease osteogenesis imperfecta. Another interesting idea, and not really interesting, kind of sad. Before we realized how radioactive radium was, it was very much involved in osteogenic tumors, osteosarcomas, because radioactive isotopes act like the stable atom. So they're easily incorporated into tissues. So ingested radium can result in osteosarcomas. So looking a little bit more at the histology of compact bone versus spongy bone. Compact bone contains osteons. So this is a basic structural unit of bone. It's concentric rings around a cylinder-like shaped structure. Each of the concentric rings is what we call lamellae. And so we have these lamellae rings that are concentrically arranged around a central canal that contains blood vessels and nerves. The individual blood vessels and nerves canals are connected via the caniculi to osteocytes. I'm gonna change it. Actually, let me say this a little differently. So the concentric lamellae are wrapped around the central canal. Within each of the lamellae, there are osteocytes living in the lacunae. If you see the little black spots here, those are lacunae with the osteocytes living in them. Well, the blood vessels and nerves in compact bone are found in the central canals. So how do osteocytes out here get nutrients? and be able to eliminate waste, because we have to do that to have a living cell. They do it because each individual lamellae and the osteocytes living within them are connected by what are called caniculi, which are little tiny tunnels. So that the lamellae, the osteocytes out here transfer waste towards the central canal. Nutrients are absorbed by moving the nutrients via the caniculi to the osteocyte where that will take it up. So we can also say that the concentric lamellae that contain the osteocytes are connected by the caniculi too. Caniculi is a connection. Allowing for the blood vessels and nerves to supply osteocytes that may be fairly far away. The central canals are connected to one another by perforating or Volkmann's canals. All of these canals, the central canals and the perforating Volkmann's canals, are lined with endosteum, just like the inner cancellous spongy bone is lined with endosteum. Within each osteon, remember that's the cylinder here. Within each osteon, the collagen fibers spiral in one direction. Here's the collagen fibers around the lamellae. And in the next concentric lamellae, they spiral, the collagen fibers spiral in the opposite directions, giving it that individual flexibility, allowing it to spiral in both ways. Blood vessels and nerves, remember, they enter through the nutrient foramina. These foramina, the nutrient foramina, are going to open into the perforating canals. And remember that the perforating canals are what connect the central canals. The innermost osteocytes within this ring, innermost osteocytes, keep giving nutrients to the outer osteocytes via the flow of waste and nutrients through the caniculi. As we've talked a lot about osteons, we've talked about how they're made of concentric lamellae. Not all compound bone is only made of osteons. The inner and outer boundaries of the compact bone are, shoot. Mom, is that you or is that one of my students? I know that's not me. Okay. Scared me. Inner and outer boundaries of the compact bone are arranged in what are called circumferential lamellae. So inner here, outer here. In between the osteons, there are interstitial lamellae, which are the remains of old osteons that have been broken down. So remember, bone is constantly being broken down and remodeled. It's constantly being built up and broken down. When it's built up, we form new osteons. And then we start breaking down some of the osteons, or we partially break them down to form new osteons. The partial remains of the old osteons that have been broken down are the interstitial lamellae. Any questions? Adjacent osteocytes communicate via gap junctions found within what? Remember what a gap junction is? That's a tunnel between two cells.
Is it B?
Say it one more time.
B as in boy.
Close, but that's actually the other name for the perforating canals that connect the central canals. And the other name for the Haversian canal is the central canal.
I'm looking at the wrong one. Sorry.
Okay. What could it be here?
Is it C?
C or D?
Oh, caniculi. Because it says canal right in the word.
It's the caniculi, because these are the ones that are spreading, where nutrients are being moved and wastes are being eliminated. This is also where the stress sensors are, because increased fluid amounts can allow for, can stimulate osteocytes to become osteoblasts again. Any questions?
No, I think the gap. Yeah, it makes sense now. The gap gave it away because the caniculi is like out in whatever land it's in. So
Right.
with the gapping, it gave it away.
Okay. Blood supply to bone. Blood periosteal arteries enter the diaphysis through the Volkmann canals. They are accompanied by periosteal veins. So here's periosteal arteries moving in through the nutrient foramina. And they're accompanied by periosteal veins. The nutrient arteries, once they enter the bone, enter through the center of the diaphysis through the nutrient foramina. And the nutrient veins enter the same way, exit the same way. Also, metaphyses and epiphyses have their own arteries and veins. So that's why we have epiphyseal veins, epiphyseal arteries, metaphyseal veins, metaphyseal arteries. innermost osteocytes around each central canal are going to receive nutrients from the blood vessels and pass them along through the caniculi to neighboring osteocytes. They also, inner osteocytes also receive waste from neighbors and convey them back to the central canal for removal by the bloodstream. An interesting method or procedure is an intraosseous fusion, which is the process of injecting directly into the marrow of the bone. This provides a non-collapsible entry point into the systemic venous system. If you think this is where all blood is coming from, is bone marrow. So this provides an easy way into the venous blood system. This or not easy way, that doesn't look like it's very comfortable, in all honesty. But this is a technique used to provide fluids and medication when intravenous access is not available or not feasible. Any questions?
Is that the same thing they would do for a bone marrow transplant?
Maybe, but I don't think they leave it open.
Gotcha.
Nerve supply to bone. So nerves accompany blood vessels that supply the bones. When you look at embryonic development, veins, nerves always follow blood vessels. So the periosteum is rich in sensory fibers. This is why breaks hurt so badly. These nerves are especially sensitive to tearing and tension. I think this is probably where growth pains comes from too. Is because, remember, this is a dense, irregular connective tissue. And you can tell how tension on that would cause pain as the bones are expanding out or elongating. Spongy or cancellous bone is composed of spicules and trabeculae with the spaces filled with bone marrow. The matrix is arranged in lamellae. If we make a cross section through one of these trabeculae means a bar. When you make a cross section through it, it looks like concentric lamellae. But there are few osteons because there's no central canals. You look here, there's no central canals. Why are there no central canals in spongy bone? Spongy bone is right next to the bone marrow, which has a lot of blood in it. Remember, it's a lot of hematopoietic tissue. So we don't really have to have central canals because everything's close to the blood anyway. That's the reason that we don't see central canals or major remember, an osteon is concentric rings around a central canal. Because we don't see central canals, we don't see a lot of osteons. Trabeculae, meaning the bars, form along the bones of stress, lines of stress. they're designed to impart strength while adding minimum weight. We'll see that later. The more surface area, also trabeculae are where we find osteoclasts because of the amount of surface area here with the trabeculae. The more surface area that's exposed. This is why cancellous or spongy bone is most affected by osteoporosis, which we'll talk about later. But because osteoclasts are involved in the development of osteoporosis, that's why we see that the most affected areas during osteoporosis is the cancellous or spongy bone and not the compact bone. A bone scan is ordered to explain skeletal pain that's often suggested to be caused by bone loss, bone infection, or bone injury that may be undetectable with a standard X-ray. It is also an important tool for detecting whether or not a cancer has spread to the bone. The way they do this is they inject a small amount of a radioactive marker and see if it concentrates in any area where there's a high bone remodeling. So you inject a little bit of this radioactive tracer and it's going to concentrate in areas where there's a high bone breaking down and building up. Exceptionally high levels. Let's, you know what? Let's stop right here. I think this is a good place to stop. Any questions?
I had a question about so how many points and how much does the lab affect our grade? Because I was like reading yesterday and
You know what? I can look and tell you. Give me a second here.
I think it's like 300 points.
Share screen.
hello.
I'm still here. Are you here? No? Did we lose each other?
Blair, you're going like in and out. Like your lips are moving, but nobody can hear you.
Oh.
Hello.
Oh,
Oh,
Now
there
we
you
can
are.
hear you. You good?
can you hear me now?
Oh, that was you. I thought it was me. Okay.
Okay. I don't think it was just you. I don't know what was going on. Horse home. Syllabus.
Oh, so it's in your syllabus. I'm sorry.
It's okay.
I was reading the lab syllabus and I was like trying to figure
yeah.
how much the lab would impact my grade.
So altogether, it's worth about 3/10 of your grade. Are you struggling in lab?
I could be doing better. I find the tests hard. I mean, I'm not failing. I just, I'm making changes to like do better on the quiz or his tests, but no, I mean, I'll be okay. I just you know, could be doing better.
I would talk to him about it too.
Okay. I mean, I'm doing all the homework and stuff. I'm not like getting A's on all the tests. I mean, some of them, but anyway, I just wanted to know how much it's going to impact my grade with you. That's all. So I see. I can figure it out.
When we submit midterms, I will reach out to him and find out just where you're sitting. I think they're due on the 18th, maybe. Somewhere right around there.
Oh, now I can hear you. I couldn't hear you for a second.
Oh, I'm sorry. They're due somewhere, when the midterms are due, which is coming up,
Yeah.
I will reach out to him and see about where you sit too.
Okay.
Okay.
Cool. Very good.
Okay.
All right. Well, you have a good day. We'll see you next week.
We'll see you next week.
All right, bye-bye.
Thank you.
Hey,
Welcome.
bye.
Thank you.
Ashley, are you still here?
Yes, I just had a quick question for
Go.
you. So basically, like I was kind of gonna ask about the same thing that Claire did. Just like she said, I'm not failing the class. It's just it's his tests are like a lot of information, and we have a week to study for it, but it's a lot all packed into like to one.
Yeah.
So like I have a good grade in your class, but in his, I think I have like a low B.
Okay.
So that's why I was wondering like how they would kind of come together.
I'll know more once I get his points added into this.
Yeah. Okay. Okay.
Yeah, I don't know right now. I would
Right.
assume if you let's say you have a low B. So what do you have in lecture? I'm gonna stop sharing.
Okay.
I'm gonna stop sharing just for a moment, but I'm definitely gonna stop recording.